keep going next and next and next, okay? So the first point to discuss today would be the point 68 of the medical syllabus. So this is the phylogeny and the development of the eye, basically. So now 2 keywords to learn and understand. Firstly, photoreception. Now photo means light and reception is basically receiving that light. So photoreception means sensation of light, being able to detect light, okay? And this is obviously what our eyes do. Then vision, it's basically a refined form of that photoreception. So the photoreception has been changed and adapted in a way that allows us to create images, okay? So in this way, our eyes and our brain work together, of course, in order for us to make sense of the light that we're receiving, and then in order for us to actually make images, okay, with our brains. Now, what are the molecular components that help with this photoreception and this vision? Well, we have opsins. And opsins are these little wavy things here that you can see within the structure. So these opsins are G protein coupled receptors, and they're able to detect photons. Photons being the light, right? Little packets of energy and the light, yeah?
Hi, sorry, I think your screen is frozen.
Okay, what are you seeing?
I just see like absolutely nothing.
Oh, okay, 1 minute. Let me stop sharing and share again. Oh, and thank you for telling me, by the way. Right. Can you guys see the screen now?
Yeah.
Yeah? Okay. All right. So I was talking about opsin. So opsin is this structure right here that you can see, this little wavy thing, okay? And they are G protein coupled receptors that are able to detect photons. Photons are little packets of energy, little packets of light, basically. Then we have the retinal. Retinal is this kind of swiggly, swirly thing inside of this thing. And the retinal is a non-protein chromophore. Chromo means color, so pigment. It's a non-protein pigment that comes from vitamin A, and it binds itself onto the opsins and allows the opsins to be photosensitive, meaning light sensitive, so that the opsins are able to detect light. Now, the retina and the opsins, when they are together, we have rhodopsin. Okay, we have this structure called rhodopsin, and we're going to talk about rhodopsin again in a little bit. But moving on now to photoreceptor cells. And this is important for you to know. We have 2 photoreceptor cells. We have the rods and we have cones. Okay. So their basic structure is similar, but they only have a difference in their outer segment. Okay. So they both have a synaptic ending, which is going to connect with the neurons and will carry signals to the thalamus, which is an area in the brain, which is going to make sense of basically the light and help us form the image. Okay. Then we have the inner segment, as you can see right here, which consists of mitochondria and endoplasmic reticulum, which will of course give us energy and also produce proteins. Then we have a connecting cilium. This is the connecting cilium. It basically connects the inner segment with the outer segment, as you can see in both these examples. Then we have the outer segment, and we said that this is different in both cones and rods, and this is because they both have different functions and they're both adapted for different reasons. Okay, they're both good for different things, basically. The outer segments are the photosensitive parts, and these are the parts that have membranous discs that come from plasma membrane. Okay, so the opsins that we spoke about here, the G protein coupled receptors and the retinals, they can be found in the outer segments. Okay, so cones, they are needed for bright light. They need bright light, and they're responsible for color vision. Whereas the rods, they're able to work in dim light, and they're responsible for night vision, but they're not very good at determining different colors on the color spectrum. So animals which are nocturnal, for example, that obviously are more so awake during the night, they hunt during the night, they do all their activities in the night, they have a lot more rods within their eyes, which will allow them to still be able to make sense of their environment when there is very, very limited amount of light available. Because as we said, this is what the rods are good for. Okay. All right. Now, the initial development. So the photoreceptors on their own are only able to detect whether there is light available. So the simplest eye form was basically just like a simple kind of like a eye spot, okay, which they would use to basically only be able to see if there was light light available, light around them, okay. Then their eyes basically, what had happened with them is that the eye basically invaginated or formed a pit or kind of a concave structure, which allowed for them to be able to sense the direction of this light. So at first, they could only sense whether there was light in their environment. And now, because of this invagination or because of this concave shape, they were now able to sense where this light was coming from, the direction of this light. And then over time in evolution, the lens developed, and the lens, of course, allows us to focus this light, okay, so that we're better able to form an image later on, okay. So the phylogeny of the eye, or the development of the eye, can be best seen in mollusks. Mollusks are the animal groups that are like, for example, the snails, the slugs. They first had pinhole eyes, which you can kind of see here. These blue structures are their pinhole eyes, okay? And then they later, when they became chordata, which were the earliest vertebrates, they would then start to use melanin as a screening pigment. Okay, so melanin basically in the iris, which would separate the photoreceptors and basically separate the amount of light that they were also getting in. So now the special adaptations in aquatic vertebrates. So we have to discuss the differences between the aquatic vertebrates and the terrestrial vertebrates, because evolution is all about talking about what changed, how did the animals have to adapt to moving from a aquatic environment to a land or terrestrial environment. So obviously when they're living in a aquatic environment, that means that they're living in water, right? So light that is passing through water, water would have a very high refractive index, right? And the fishes and the amphibians that are living in water, they would need to have lenses that could accommodate this. Lenses which are going to basically be spherical in shape, okay? And this would be so that they'd be able to get the most amount of light. But you have to remember that aquatic vertebrates, they don't have the best sense of vision, okay. Vision would improve as you go up, okay, as you go up the evolution. So they developed spherical lenses in order to accommodate for this refractive index, or the high refractive index of the water. And another thing that changed or happened in aquatic vertebrates is that they were able to move their lenses forward or backwards, okay, and this again would control the amount of, or would basically focus the light that they were getting in. Okay, and due to them being able to move the lens forward or backwards, they also developed these specific muscles that would adjust the lens according to what they needed. Okay, and the environmental advantage is that aquatic vertebrates, they don't experience issues with dry eyes, of course, because they are in an aquatic environment, okay? And for this reason, they don't need protective features. And these protective features are things, for example, such as lacrimation. Does anybody know what lacrimation is?
Right?
Crying, exactly. So basically, aquatic vertebrates, they don't need to produce tears. And I don't mean tears in the sense of like the emotional tears, but I mean tears in the sense of like just the liquid, the fluids. These aquatic vertebrates, they don't need to produce these tears because they're, of course, living in an aquatic environment. Their eyes are constantly lubricated by the water that is around them. Okay. And it's important that the eyes are having some lubrication because, of course, the eyes, as we mentioned, the lenses are moving forwards or backwards. And if they're moving forwards and backwards and there is no sense of, there's nothing to lubricate, then there's going to be some friction, and this is going to cause damage to their eyes. Okay, so this is why it's important to not have dry eyes. Now, how did this change in terrestrial vertebrates? Well, now that they started living on land, they had to adjust to the air, right? They're not living in water anymore. And air, compared to water, water is more dense and air is less dense, right? So the other thing that changed is that their lens became a bit more flat because they're not dealing with that whole high maximum refractive index of the water anymore. Now that they're living on land, amphibians and the snakes, they use the lens in order to. They basically move the lens in order to see things properly, whereas the amniotes, so excluding snakes, so all the other reptiles and snakes, reptiles and birds, including snakes, and then mammals, they change the shape of their lens, just like we do, just as humans do. We change the shape of our lens. So the fishes, or the aquatic vertebrates, the amphibians, and the fishes, they move their lenses backwards and forwards, whereas we change the shape of our lenses. Okay, and this is, of course, again, to control the focus of the light that's coming in. Now, because we change the shape of our lenses, we are vulnerable to something called presbyopia. And presbyopia is age-related farsightedness. This basically means that over time, because we keep changing the shape of our lens, of course, this is involuntary and controlled by the autonomic nervous system, because we're constantly changing the shape of our lenses, eventually what will happen is that kind of flexibility in being able to change the lens is going to decrease. Those muscles that are helping to do that, they're going to start to deteriorate. And eventually, because of that, what's going to happen is we're going to develop, or some of us, some humans or some animals which are able to change the shape of their lens, are going to develop farsightedness over time, meaning that they won't be able to see things close to them. They won't, like, the focus of their eye is not going to be as good, where they're not able to focus properly on things that are close to them, close in distance. Okay, so now the protection in air environments. So we said that it's very important that the eyes are lubricated and that they have some level of moisture. And due to this, the terrestrial vertebrates, they developed the lacrimal apparatus. As Mohammed told us, this is basically to produce the tears, okay, to lubricate the eye and, of course, protect against friction and damage. And then we also developed eyelids, and this was to protect from debris, things kind of floating in the air, and to keep the moisture within the eyes. We also developed eyelashes. So camels, for example, or other desert animals have very long eyelashes, and this is to, of course, protect their eyes from sand debris that is constantly floating in the air. Okay. Now, something that is very unique to humans is the emotional lacrimation. So we are the only species that is able to cry due to a response to an emotional stimuli, basically. Okay, no other animal is able to cry because of an emotional stimuli, as far as we know. Okay, they are still able to produce tears, of course, but this is to protect the eye from friction. All right. Any questions so far? No. Okay. Now, amphibians is the most interesting group because what's it called? In their early forms, in their early development, of course, they are still living in water. And as they become adults and they experience metamorphosis, they start to live on land more predominantly, basically. So due to this, their eye structure basically changes. In the beginning, they have eyes that are suited for underwater vision. And the eyes that are suited for underwater vision are, of course, the spherical shape that we were talking about earlier, because this spherical shape is able to accommodate for that high refractive index of the water. But then, as these amphibians go through metamorphosis and as they become adults, and as they start to live more on land, their lens, of course, is going to become a bit more flat. They don't need the spherical lens anymore. And this flattened lens is now going to be able to allow them to focus this light in less dense medium, which is the air, right? And they, of course, also develop their eyelids as they become adults, again, to protect from that loss of water within the eyes and to also protect from floating debris in the air. Okay, now we have another thing that's quite interesting. We've got something called the tapetum lucidium here. Now, tapetum lucidium is something that can only be seen in nocturnal animals. So, for example, cats, okay? And you've probably seen this before, where you've taken a picture with flash of a cat before, and then you see like this blue or kind of green yellow glow in their eyes. And this is because of their tapetum lucidium. Now, the reason they have a tapetum lucidium and we don't is because technically we're not nocturnal animals, okay? We're diurnal animals, meaning that we basically do, of course, all of our activities and everything usually during the day, and we sleep during the night, which is different to cats. So basically, because of this, because we're not nocturnal animals, we don't have this tapetum lucidium. As the tapetum lucidium, what it actually does is it maximizes the amount of light that the cat eye actually receives. Okay, so if we just zoom in a bit more, because I'm kind of blind, I'll show you something. Okay, so the way that vision normally works, or the way that we receive light, is of course light travels from our pupils, and then our lenses basically focuses that light and sends it to our retina. Inside our retina, we have our photoreceptors. Remember these cells that we were talking about earlier, our rods and cones? Our rods and cones are actually within our retina. Okay, and remember that we said that the rods are important for basically being able to detect light in very dim circumstances when there isn't a lot of light. Well, the cat's eye in their retina, firstly, they have a lot of these rods. Okay, they might not have as many cones. They could have some, but they won't have as many cones as they will have rods in their retina. Anyways, so once this light is focused from the lens and then is sent to the back of the eye where the retina is, it will just go straight into the retina. The photoreceptors will detect this light, and then they'll basically send, of course, the signal to the optic nerves, which is at the back of the eye. And then this was going to send a signal to the brain, and then the brain is obviously going to form that image, right? But in the cat eye, what actually happens, or other animals which have the tapetum lucidium, is that the light enters through the pupil and then is focused by the lens, hits the retina where the photoreceptors are, but it doesn't stop here, okay? Because it hits this membrane, this tapetum lucidium, okay? And then when it hits the tapetum lucidium bounces that light back into the retina so that that light is now magnified or multiplied, okay? So imagine you have one ray of light that hits the retina, then it's hitting the tapetum lucidium, then it goes back into the retina. So it goes, light basically now is going into the retina twice, okay? Which basically allows these nocturnal animals to now be able to see in the dark because they had very, very little light available, but they were able to magnify that light. I hope that makes sense. Okay. Yes. All right. Now, another thing to mention is you may have taken pictures of maybe your friends, family members, or even yourself with flash before, and you may have noticed like a red eye effect. Okay. And this red eye effect happens because we have this structure here called the choroid. Okay, most animals they have this structure here, and this choroid basically is our innervation for our eyes as it provides our blood vessels. Okay, so when you're taking a picture of somebody's eyes and there's like low light, and then all of a sudden there's that flash, right? You're seeing that redness. That redness that you're seeing is actually the blood vessels which are in the choroid. Okay, and that is all there is to this slide. Okay. Now the color vision. So the opsins, these are the proteins in the photoreceptors that absorb the light at different wavelengths. As you know, colors come at different wavelengths. And this allows us to see these different colors because of the opsins that we have. So initially, the ancestral vertebrates, they only had two opsins, and their retina was made only of cones. Okay, so because it was only made of cones at the time, this meant that their vision was best when there was a lot of light available. So in the dark, they could see absolutely nothing because they didn't have any rods. Okay, so there was nothing, there was no photoreceptor cell which was helping them with dim light situations because they only had the cones. But because of the whole genome duplication, this led to making more opsins, and some of these opsins basically became rhodopsin. Remember the rhodopsin were the opsins which were combined with the retinal. Okay, and these rhodopsins can be found in large amounts inside of the rods. The rods have rhodopsin, and this rhodopsin allows them to basically be used in very dim light conditions, so that these animals now that have a lot of rhodopsin, they are able to see things now in the dark. Okay. Now we're at the end of the eye, so the development of the eye. How does the eye develop? So firstly, the eye actually develops from the ectoderm. The ectoderm helps a lot, okay? So firstly, what's going to happen is the eye is going to develop from the forebrain, from a region called the diencephalon. Okay, this diencephalon is going to make two protrusions. It's going to make two protrusions right here. So this area here, which is the optic vesicle, and this area here, which is the other side of the optic vesicle. Okay, we're making these two protrusions. Okay, then these two optic vesicles, they're going to extend outwards until they contact the ectoderm, which is here. Okay, they're going to contact the ectoderm, the surface ectoderm. And then the optic vesicles are going to, of course, they are connecting a little bit with the ectoderm, and then the cells within the ectoderm are then going to become a bit thicker. They're going to cause the ectoderm to become thicker, and as the ectoderm thickens, it forms the lens placode. The lens placode is basically the first or the original part of the lens. It's what's going to form the lens later. Okay, so the ectoderm gives rise to the lens, and because of signals from the ectoderm, we also form our optic vesicles. These optic vesicles are later going to form our optic cup, as you can see right here, and this optic cup is then going to give rise to this narrowing, which is going to be the optic stalk, and inside the optic stalk, we're going to have our optic nerve. You don't need to know how the optic nerve develops. What you need to know is that the ectoderm gives rise to the lens, okay, and the ectoderm also signals for the optic vesicles to be created and for them to form these pits and for them to become rounded, okay. At the same time, the retina forms, as you can see, and the retina is eventually going to move to the back of the eye as the lenses, as you can see right here, the lens, it's going to detach itself. It's going to form a pit first. As you can see, the ectoderm is forming a pit. As it forms this pit, it's eventually going to separate from the ectoderm and round off, and it's going to form our lens vesicle, okay? And as this happens, or after this happens, what's going to happen is that the other part of the ectoderm is now also going to form the cornea. And the cornea is our transparent and protective layer, which is protecting our lens and the rest of our eye. Okay? Does anybody have any questions so far before we move on? No? Okay. All right, now we're going to talk about the phylogeny and the development of the digestive system. So as I said before, we first start off with the overview of what each system is, what it's important for, what it does, etc. Okay, so of course, as you all know, the digestive system is important for us to ingest, so basically eat and digest and break down our food. So food goes through the mouth, it's broken down first in the mouth, and then it's broken down further as it goes down the digestive tracts, and then eventually goes into the small intestines, where nutrients from the food are going to be absorbed into our bloodstream. And then, of course, our blood is going to carry these nutrients to the cells and the tissues that need them. Okay. The digestive system is also important for the removal of undigested remnants. So basically, waste products or things which our body doesn't really need anymore, or things which can't really be digested properly, are going to be eliminated from the body through the anus or the cloaca. The cloaca is a structure that reptiles have, and it's kind of similar to our to the anus. Okay. So, what are the parts of the digestive system? Well, we have the major parts and we have the accessory organs. The accessory organs are basically our helper organs, where the major subdivisions are the main parts of the digestive system. So, of course, we have the oral cavity, so basically our mouth. We have the pharynx at the back of our oral cavity, which is going to help to push the food down into the esophagus, which is our food pipe. And of course, our esophagus is that link between the mouth and the stomach. And then the food goes into our stomach, where it's broken down even more because of our gastric acids. And then the food travels into our intestines, the small and the large, where in the large, of course, water is taken from the food or reabsorbed, and in the small, the nutrients are absorbed basically into the blood. And then the anus or the cloaca is the end point of our digestive system, where we excrete the waste products. Then the accessory organs, of course, as we said, these are the organs which are helping, or the parts of the digestive system which are helping. So this would be the tongue, which will allow us to push our food down or move our food downwards. Our teeth, which are important for breaking down the food into smaller components. Our salivary glands, which contain our enzymes, of course, which are going to break down the foods even more. Our liver, which are also going to basically contribute to breaking down the foods a bit more. The gallbladder, the same, and the pancreas, which basically releases our hormones, for example, our insulin, which is going to control the glucose levels within our blood, our glucose levels within our blood. This is something that they also mention in the lecture slides. So if you mention it in the exam, it will be very good. But basically, the enzymes that we have that are involved in digestion are initially going to be called zymogens or proenzymes. Okay. Now, these are contained by the glands, and they are only activated once they leave the glands. This is because if they were to be activated whilst still in the glands, they could damage the glands. Okay, so these zymogens are only activated once they've left the glands and once there is a change within their environment which causes them to become activated. And this change is usually due to proteolysis, which is the breakdown of proteins. So an example of this would be pepsinogen. Pepsinogen is a zymogen. So when it's inactive, it's called pepsinogen, as you can see right here. It becomes active and starts working in the stomach once hydrochloric acid is released by the stomach. Okay, once this hydrochloric acid is released, this activates the pepsinogen and allows it to become the active form pepsin, which is going to break down the proteins, right? And looking at this picture, we can kind of understand how that happens. So as you know, hydrochloric acid has a low pH, okay? Once there is a low pH, this masking sequence, which is in the structure of the pepsinogen, okay, this structure right here, will be removed, and this will expose the active site of the enzyme. What does the active site of the enzyme do? Can somebody tell me? What's the purpose of the active site of the enzymes?
To fit its substrate so it can function.
Sorry, you said this so fast. What? Sorry?
I said the purpose of the active site is so the substrate can fit in so it can do its function.
Yes, exactly. The active site is that area where the substrate is going to go in, and then the enzyme is going to be able to break it down. Guys, can you still see my screen, by the way?
Yep.
Yeah, okay, 1 minute. Okay, so very good. So moving on. Now, amylase is actually very interesting also, because amylase was not an enzyme that existed earlier in time, and amylase is it's a newer enzyme basically. And amylase is basically produced by the salivary glands in the mouth. Sorry, the amylase is produced by the salivary glands in the mouth, and they're important for breaking down the starch. Okay, And starch is a newer thing that we started to eat with the help of, or with the new formation of agriculture, with the new existence of agriculture. Once we started farming and we started growing things like potatoes, and we started growing certain rice plants, etc. Once we started growing these things and we started eating these things, is when the amylase enzyme started to form in order to help us basically break down the starch products. And of course, starch can be found in all of these foods, so bread, rice, cereals, potatoes, etc. Okay, so other animals, not just mammals, eat starch. Other animals may eat starch too, so dogs, rats, pigs, mice, and they all develop this amylase enzyme as a result of this. Okay, so the evolution of the digestive system in vertebrates now. So this is the second part. And this was when I was talking about the phylogeny. So as you may remember from last week, we said that the first vertebrates, the first animals, they weren't really predators, right? They were bottom feeders, filter feeding animals, which lived at the bottom of the ocean, and they basically used to filter water in order to get their nutrients. Okay, so because of this, they basically had a large pharynx, which had numerous slits in the pharynx in order for them to be able to trap the food particles from the ingested seawater. They also had cilia, cilia basically is these structures that look like hair, which basically waft and they move basically in a sort of forwards and backwards motion. And these cilia, they had a lot of them, as these cilia would basically move the nutrients basically from the water and through their digestive tracts. Okay, as the vertebrates evolved and they became larger and they became more predators, they basically developed more of a muscular pharynx, which replaced the need for the cilia. We didn't need the cilia anymore. We got the muscular pharynx. As I mentioned before, the pharynx is important as it basically allows us to push our food down from our mouth into our esophagus. Okay, so this transition going from the cilia to a muscular pharynx basically helped in us to basically go from filter feeding to active predation. Then, of course, as you know, we started to develop the jawed vertebrates, and these are vertebrates with jaws, of course, and this again helped with the predation. It allowed these animals to now become better predators as they had stronger jaws, cartilaginous jaws, jaws that had very sharp teeth, for example. And these are basically, in short, these are the things that changed within the digestive system of the vertebrates. Okay, so here is the pharynx with slits that we were talking about. At first, we said that these animals, the early chordata, they were filter feeding animals where they would basically take in water and then remove the nutrients from that water, and they would take in that water through their pharynx that had slits right here. And then they would have cilia within their body, which would basically move those nutrients down the digestive tract, and then they would eventually excrete them. Okay. So now the teeth evolution and the oral anatomy in cartilaginous fishes. Now cartilaginous fishes like sharks, they have something called placoid scales. Okay, right here. They're placoid scales. And the placoid scales are very interesting because they share a similar structure to our teeth, right? Because our teeth, and I don't remember if we mentioned this last week, we may have on the Sunday, but the teeth basically have this structure of having a outer enamel, which is that harder surface that will protect the tooth. And then we have our dentin, which is that softer area on the inside that is innervated by the pulp, which is in the middle. And this pulp has got all the blood vessels and all the nerves, which are important to sustain the dentin portion of the tooth. Now, this exact structure that we have in our teeth, the sharks or the cartilaginous fishes have in their placoid scales, which basically cover their skin. Okay. The shark teeth are conical in shape, meaning they're like cones, and their oral cavity seems to be deeper because when they open their mouth, we can actually see deep into their pharynx. Okay. the tongue in the fishes is immotile, it doesn't really move, and it's crescent shaped and covers the hyoid skeleton, which is at the back of the mouth. Okay. Now, unlike cyclostomes are agnata. Can anybody remember what agnata is? What is an agnata?
Jawless
Fish?
fish.
Yes, exactly, jawless fishes. So cyclostomes are also called agnata or jawless fishes. So unlike jawless fishes, the cartilaginous fishes, they have distinct stomach. So a stomach that actually looks like a stomach, okay? And this stomach has developed and basically become a storage site for their food and also a macerating site. By macerating, we basically mean an area that is breaking down the food even more. Okay, so their stomach is basically churning and breaking down that food even more and also storing that food. And then the stomach later on started to develop hydrochloric acid and also the enzyme pepsin to break down these proteins. Okay, but this was a later adaptation. Okay. Now, what changed in the amphibians? Well, since the amphibians were the first land animals, they started to eat different things now. Okay. At first, these amphibians they were very large and they were eating fishes. Okay. But over time, they started to become insectivorous and they became smaller, meaning that now their diet only consists of insects. Okay. But in order to really digest these insects, they had to develop these salivary glands for two reasons. Number one, when the fishes basically are in water and they are eating their food, for example, when a shark is in water and it's eating other fishes, it's able to very easily eat that food without choking on it, right? Because it has a lot of water, it's taking in a lot of water at the same time. But once these amphibians transition to land, there is no water on land, right? It's just, I mean, there's water on land, but like their environment is mostly air, right? So they developed these salivary glands to basically lubricate their food a bit more so that they'd be able to swallow their food, okay? So that their food would not get stuck they would not get stuck basically and they wouldn't choke essentially, okay? So they developed the salivary glands for moisturizing this food and also to basically have these enzymes which would break down their food a lot easier also, a lot better. Now, during metamorphosis, or just before metamorphosis actually, metamorphosis is when they obviously go from their embryo stages, larva stages, and to their adult stages. But whilst they are still larvae and they are living in water, when they are tadpoles, for example, they still have their pharyngeal slits, okay? In the beginning, they still have their pharyngeal slits because the way that they're getting their nutrients is they are still, at that point in time, they are still basically removing nutrients from the water through their pharyngeal slits, right? But as they become adults and as they're going to start to be living primarily on land, they don't need these pharyngeal slits anymore. So the pharyngeal slits are going to close, okay? And when they close, the pharynx is going to, of course, become a bit more muscular, the esophagus is going to become longer, and they also developed the intestines. So the intestines were seen for the first time in the amphibians. This would be, of course, the small and the large intestine. Okay. Now, the reptiles and other amniotes. So if you remember from last week, we said that the reptiles had to adapt to living life away from water. Okay, so they'd live very far away from water. Of course, some of them may still live close to the water, but most of them would live very far away from the water, and they would not know when their next water source would be, when they would next come across their next water source, right? And for this reason, it's very important that they retain water and that they don't lose water unnecessarily. Okay, and due to this, the reptiles and other amniotes, they developed the cecum right here, which is basically the beginning part of the large intestine. And this cecum here is important for reabsorbing water. Okay, and these reptiles, they needed this, and these amniotes, they need this so that they are not losing too much water and they are still keeping some of that water within their body, which of course, as you know, water is needed for many things. For example, for maintaining the water electrolyte balance. Okay. Now, the mammals, what changed with the mammals? So the mammals, their our mouth and our oral cavity developed a bit more as our lips became more motile and we're able to move our lips a bit better, and our cheeks became more muscular. Okay, and this was for two reasons. Reason one is suckling, and reason two is mastication. Now, suckling is basically receiving milk from the mammary glands. Okay, so basically sucking that milk. That's what suckling is. So we had to do this because, of course, we said mammals, they get their name from the mammary gland because the mammary gland appeared for the first time in mammals, or mammals are the only animals to have the mammary glands. What do the mammary glands do? What's the purpose of the mammary glands, guys? I would like to ask Labour the question. Labour, what do the mammary glands do?
They pass antibodies like IgA and nutrients as well.
Exactly. So they contain the milk, which contain these antibodies that you mentioned. Exactly. So the mammary glands, they have the milk, of course. So because of this, because we needed to receive these nutrients from our mothers, the cheeks became more muscular and the lips became more motile. And they also became more motile and more muscular because of mastication also. And mastication is that chewing, that mechanical breaking down of the food. We also developed a secondary palate for the first time. Here is the primary palate that you can see, and here is the secondary palate. Now, mammals are the first animals to have this secondary palate. All the other vertebrates before, they only had the primary palate. But the mammals developed the secondary palate because, of course, mammals are larger animals, usually, and they need more nutrients, so they're going to eat more animals, and they need the secondary palate in order to help push that food down to the, or basically back to the pharynx and then down into the esophagus. So that's why we developed this secondary palate. Okay, now the tongue functions and the salivary glands. So the tongue has got primary roles in food manipulation and taste perception, as we spoke about last week, where we spoke about the different taste buds that the tongue has. Remember that we said that in the evolution of the tongue, or the evolution of the taste buds, we developed the bitter taste bud, or the animals developed the bitter taste bud when they ate certain plants, because this let them know, okay, this tastes bad, I'm going to avoid this. And usually the things that tasted bad were things which were toxic to them. Okay, so the tongue has got that function, of course. But in some mammals, like felines, like cats, for example, they have these papillae, or specialized structures on their tongues. So if you ever look at a cat's tongue, you'll see like these like white rows on their tongue, and they're basically these hard structures which can remove meat from the bones of the animals that they're eating, or they can remove basically dirt from their fur. Okay. And of course, in humans, our tongue is also very important for speech. Now, the salivary glands, mammals have four main salivary glands: the parotoid, the mandibular, which is also called the submandibular in humans, the sublingual, and the zygomatic, which we as humans don't have. And of course, these salivary glands are important for producing the enzymes like amylase, which we need for starch digestion. Now, we have vampire bats, which basically what they do is they feed on blood. Their main diet, or their only diet, is blood. So they will go hunting for animals, whether dead or alive. Usually the animals would be alive. So they would go and they would basically attach themselves to an animal and essentially suck out the blood of that animal. And they're quite evil, okay? Because when you cut yourself, when you cut yourself, right? Your body will do everything that it can to send, of course, the platelets there, the fibrinogen there, the blood clotting factors, basically, in order to create that blood clot, right? In order to stop the bleeding, right? And the body does this so that pathogens don't come in, correct? So the vampire bats are evil in the sense that they have something called draculin, and draculin is a anticoagulant glycoprotein, meaning that when these vampire bats, they attach themselves to an animal and they try to suck out the blood, when they've pierced the skin and the animal is bleeding, they release these enzymes, or not these enzymes, but they'll release this protein, this glycoprotein, And this glycoprotein is basically going to slow down or basically stop that blood clotting process that the victim animal would be trying to do, basically, so that the animal will just continue to bleed out, and then the vampire bat would get as much blood from it as possible. So that's why I said that the vampire bats are kind of evil in this sense. Okay, so the evolution of teeth then. So sharks have a unique dental system where they are constantly able to replace their teeth. You know, in humans, we have our milk teeth, and these basically fall out around the ages of 6 to 7, and then we develop our more permanent teeth, our adult teeth, right? But sharks, they don't have this. Sharks are constantly renewing their teeth, constantly getting new and new and new teeth, okay? And this is basically to help them, of course, with their digestion and basically eating their prey and breaking down their prey and so on. Mammals have got heterodonty. Heterodonty means that we have different types of teeth, and this is because we are omnivorous, meaning that we are eating plants and we are also eating animals. So we have teeth to help us with our different diets. So these are the main teeth that we've got. We have our incisors, which are our front teeth, our canines on the side, our premolars, and then, of course, our molars. And our premolars and our molars are adapted for breaking down plant-based foods, whereas our canines and our incisors are for like ripping apart flesh when we're eating other animals. Okay, so this is something that is unique to mammals. Okay, that we have all these different type of teeth, whereas other animals or other mammals, okay, they may have other type of teeth where, for example, if their diet is mainly just plants, then they'll have more like flatter, thicker teeth, like the premolars or the molars. And then, of course, if they are more carnivorous and they only eat meats, for example, sharks, then they're just going to have these incisors and these canines. They're just going to have the sharp teeth, basically. Okay. So mammals have evolved specialized teeth to accommodate endothermy. Can somebody tell me what endothermy meant? Okay, I am going to ask Abdul. Abdul, do you remember what endothermy and ectothermy was? No? Okay, Victoria, do you remember what is endothermy?
I think it's when you're able to regulate your own temperature.
Yes, exactly. Exactly. It's being able to regulate the temperature. So which animals are able to regulate their own body temperature? And I will ask Angela, which animals are able to regulate their body temperature?
I'm not sure, but I think it's reptiles, maybe amphibians, fishes.
Oh, okay. No, they are not able to regulate their temperature, actually. Those are the animals which are not able to regulate their own body temperature, as they depend on the sun for heat. Okay? So
You said
reptiles,
endothermy or ectothermy?
endothermy. Endothermy.
Okay.
Yeah, the endothermy ones are the ones that are able to regulate their own body temperature. So these are going to be the mammals and the birds. Okay, those are the two animal groups that are able to regulate their own body temperature. Now, the interesting thing about endothermies is that these animal groups, they usually require a lot more food. Okay, they require a lot more nutrients in order to basically control their body temperature. Okay, so they basically developed intensive chewing and good occlusion, so that they're able to really chew on their food and break it down and then swallow it. Okay, and yes, that's all we need to know for this. Now, the development of the digestive system. So this is the third part of the question. How did the digestive system develop? Well, firstly, it comes from the endoderm. Okay, and it forms a hollow longitudinal tube that is known as the primitive gut or the archenteron. Okay, then the mesoderm, which is of course above the endoderm is going to give the structural support, and then the ectodermal epithelium, which is even more above this area, is going to cover the terminal compartment, so the end of this longitudinal tube. Okay, chordata are deuterostomes. We mentioned last week that deuterostomes are animals that develop their anus first. Okay, so their blaster port basically becomes their anus first. Do you guys remember the name of the groups? Or what's the opposite? What was the opposite of deuterostomes? The deuterostomes are animals which develop their anus first. What was the animal that what were the animal groups that developed something else?
Protostomes,
Protostomes, and they develop what first?
first the mouth, second the anus.
I'm sorry, what?
First was the mouth and then the anus.
Yes, exactly. Very good. Very good. So protostomes, they develop their mouth first. Okay. Where are we? Yes. Okay. So in mammals, the digestive system starts as a blind-ended gut tube. So we basically form a tube when a portion of the yolk sac incorporates into the embryo. Okay. So the yolk sac basically, what does it do? What is the purpose of the yolk sac in the embryo? And this is something you should know from cytology. So what does the yolk sac do? Why do we have a yolk sac in the embryo? Mohammed, do you know?
Is it to give the rest of the nutrients?
Exactly. To provide the nutrients for the embryo. Very good. Okay. You guys can still see the screen, right?
Yes.
Okay, cool. So the gut, basically, the gut tube develops from the yolk sac. Okay, the yolk sac is of course going to give the nutrients. Then the yolk sac is going to connect to the vitelline duct temporarily. Here is the vitelline duct. Okay. All right. So the accessory digestive organs develop as outpouchings from the primitive gut. So they're basically going to come from this primitive gut. The accessory organs were, of course, what we mentioned before. So the tongue, for example, the pancreas, the gallbladder, the liver, all of these are the accessory organs. The intestines, they initially go into the umbilical cord first through something called umbilical herniation. This is a normal thing. So they go through the umbilical cord first, and then later on, as they develop more, they go back into the abdominal cavity where they stay. Okay, so here is the foregut, here this area, and then here is the midgut, and of course here is the hindgut. So the hindgut is going to be responsible for forming the cloaca or the anus later, right? Or I mean, that's what it forms first. Okay, and then the midgut is responsible for forming the small intestines and the large intestines, and then the foregut here, as you can see, is starting to take the shape a little bit of the stomach, as it's going to start to form the stomach. Okay, so that was the end of this topic. So if you're a dental student and you don't want to stay, then you can go, because now we're done with the dental topics. But before you go, do you have any questions? Does anybody have any questions? No. Okay, so we're going to go to the next syllabus point, which is number 70, about the circulatory system. Okay, so we first have to discuss what the circulatory system is like in the invertebrate animals. Okay, so firstly, the invertebrate animals, they will either have a open circulatory system or a closed circulatory system. But when we're talking about evolution, we're going to go from having an open circulatory system to having a closed circulatory system. The best type of circulatory system for us is a closed one, okay? And the way that you can understand what the difference is from learning, or from remembering actually, parts of physics. Do you remember when we had to learn about circuit systems, and we learned that the circuit would work if it's kind of closed, right? And there was no gaps or no breaks. Well, that is a closed circulatory system, whereas an open circulatory system has got gaps everywhere. Okay, now the invertebrates, they lack oxygen transporting cells. Which cells transport oxygen in our bodies?
Red blood cells.
Exactly, very good. Red blood cells. So the invertebrates, they don't have red blood cells. They usually have an open circulatory system, and usually what happens is that their blood is actually able to leave their vessels. You know their blood vessels, their veins and their arteries? The blood is actually able to leave them, okay? And then it goes through sinuses around the organs. Sinuses are basically gaps or spaces in between the organs, okay? And this is good for the small body size that these invertebrates have, for example, for insects, okay? They need this. This is good for them. Okay. Now, annelids, they have a closed circulatory system. Now, annelids were the segmented worms, right? So they have a closed circulatory system. Their blood has hemoglobin in it, okay? And this hemoglobin is found inside of their plasma, dissolved in their plasma. Okay, their blood works basically both as blood and lymph, okay, where they're basically carrying the liquid, they're carrying lymph basically around the body. And they're able to transport nutrients around the body, but they're not able to transport gases around the body. Okay, all right. Now, tunicates are very important to mention when it comes to the evolution of the circulatory system, because tunicates are invertebrates. They are not vertebrates, but they are closely related to vertebrates because they were the first animal group to form a distinctive heart, meaning something that resembles a heart, which is this here, okay? So they formed a single-chambered, tube-shaped heart, okay? They don't have a vascular network, but they're very, very simple. They just have this tubular heart. Okay. Now, the vertebrates, they formed, of course, eventually a multi-chambered heart. So the first animal or the first vertebrate to do this would be the agnatha. Okay. So lamprey is an example of a agnatha. Okay. So agnathas, they formed a multi-chambered heart in the sense that they had an atrium and one ventricle. All right. Because of this, whenever we have a multi-chambered heart, we can actually call it a cardiovascular system. Okay, because there's some sort of organization happening here. So they had a sinus venosus right here, which would collect blood from the veins and would also act as the heart's pacemaker. And the blood would go through the thin-walled atrium. The atrium would be thin-walled, and then would travel to the thick-walled ventricle. Then the ventricle would pump the blood to the conus arteriosus, and this would, of course, send this blood to the to the cells and the tissues that would need it. And as we said, the early vertebrates, so the agnatha, have a two-chambered heart. Okay. Now, the heart in fishes, so the way that their circulation works is, firstly, they use their gills. We said that they take in water from their gills. No, they take in water from their mouths, and then this water passes through their gills, and then their gills are able to extract oxygen from that water. So you can see that here. So they're extracting oxygen from that water, and their gills are connecting to the efferent branchial artery. So this is carrying oxygenated blood, or sorry, oxygenated blood basically, and it goes through the dorsal aorta and is then sent to the tissues. Okay, then eventually the tissues are going to use up this oxygen, and the blood is going to travel back to the sinus venosus, then to the atrium, then to the ventricle, then to the bulbous area, then to the ventral aorta, through the afferent branchial arteries, and back into the gills to sort of be renewed again with oxygen. Okay, and that is how their circulation works when there are fishes that are using gills. Okay. Now, the aortic arches are also very, very important to speak about. So, the heart, of course, normally has a aorta, right? And this is the thickest and the biggest artery, right? So, in evolution, this aorta has split up in different branches as it carries the blood to the head, to the neck, and to the neck. Okay. So, what changed in gill breathing fishes? Well, they lost arch 1 and arch 2. So you can see that in bony fishes. They lost the aortic arch 1 and the aortic arch 2, they remained with the aortic arch 3, 4, 5, and 6. Okay, whereas the cartilaginous fishes, they only lost the aortic arch 1, and they stayed with the aortic arch 2. The aortic arch 2 remained, they're 3, 4, and 5, and 6. Okay, so the arch 3 actually supplies oxygenated blood to the head through the carotid arteries. Okay, now the heart and circulation in lungfish. These were the first fish to develop lungs. Okay, so even though they had lungs now, they would still use their gills to some extent. But as the lungs developed in these lungfish, it meant that the double circulatory system also developed. Now, the double circulatory system, you should already know, is the systemic and the pulmonary, where the blood is traveling to the heart and is also traveling to the lungs. Okay. So because of this system, the sixth aortic arch basically formed the pulmonary artery, okay? And the oxygen-rich blood would return to the heart and then be pumped to the head. And in this, actually, in lung breathing, there was still some mixing of blood happening, okay? And you're going to realize that as we're going up in evolution, this mixing of blood is going to be stopped, okay? Initially, it's not a problem because these animals are smaller, okay, and they actually benefit from having mixing of blood initially. But then as these animals become larger and they become humans, for example, humans, they don't have mixing of that blood, okay, and this is because of our metabolic needs. Okay, so the aortic arches in amphibians, there was connections that were lost between arch 3 and arch 4, okay, not arch 4, sorry, arch 6. There were connections that were lost between arch 3 and arch 6, and the carotid duct, which connects arch 4 and arch 3, they basically became reduced. Now, urodela and anura, you may be wondering what these two things are. Urodela are the salamanders, okay, and then the anura are the frogs, okay. So these are the aortic arches within the salamander and the frog. Okay, the amphibians were the first to actually develop a 3-chambered heart. Before that, we only had 1 atrium and 1 ventricle. Now, with the amphibians, we have 2 atria and 1 ventricle. The atria would receive the blood, the venous blood, and the left side would basically then receive the oxygenated blood. Now, arch 3 would supply the oxygenated blood to the head through the carotid. Then arch 4 is going to bring mostly arterial blood to the body through the systemic circulation. And then arch 6 is going to form the pulmonary artery. And, of course, going to contribute to the pulmonary circulation. Okay, so what changed with the reptiles? So the reptiles still have a 3-chambered heart where they have 2 atria and 1 partially divided ventricle. There is a mixing still of the oxygenated and deoxygenated blood because the ventricle is not split completely. But in crocodiles, they have a 4-chambered heart, okay? And this is normal for them. So it's acceptable for reptiles, except for crocodiles, to have some mixing of the oxygenated and deoxygenated bloods because their metabolic needs are lower than mammals and birds, okay? Their metabolic needs are lower because these reptiles are ectothermic, okay? They don't need as much nutrients because they don't regulate their own body temperature, right? But mammals and birds, we regulate our own body temperature, so we have more of a metabolic need, and due to this, we don't have mixing of our oxygenated and deoxygenated blood, okay? Another interesting thing about the crocodiles is they have something called a foramen penisa, and this foramen penisa is a small opening, okay? And this allows the crocodiles to, when they're diving in water, they're able to hold their breath, and at the same time, they're also able to move the direction of their blood, or basically they're able to adjust the flow of their blood so that the blood doesn't go to the lungs, and in this way, they're able to sort of conserve oxygen. Okay, they bypass the lungs or bypass certain tissues so that they're able to conserve oxygen. Okay, and they are able to do this, or this is helpful to them when they are diving deep into the water. Okay, so changes in endotherms. Well, the ventricle septum basically developed after post-embryonic. So basically, after them being an embryo, the ventricular septum developed, which basically separated the ventricle a little bit. And then the heart became four-chambered, of course, and the major arteries were the pulmonary trunk and the aorta. So what happened to the sinus venous that we mentioned before? That was kind of the pacemaker of the heart. Well, the sinus venosus basically fused together with and became part of the right atrium. It stopped being its own distinct chamber when the animals transitioned to becoming the endotherms. Now, the red blood cells in vertebrates is also very different. So non-mammal vertebrates, their erythrocytes are elliptical shaped, so they're like elongated, and they have a nuclei and organelles, right? So us as humans, our red blood cells, we don't have nucleuses within our red blood cells. Can somebody tell me why? Why don't we have a nucleus in our red blood cells?
So that
Is
we
it so that
take
they can be
more oxygen.
biconcave?
So that they can be biconcave, yes. And why is this important? But I mean, there are other reasons also, but yes, tell me. So why is this important? Why do they have to be a biconcave shape?
So they can fit into like smaller capillaries and other stuff.
Exactly. Can anybody tell me any other reason for why red blood cells don't have a nucleus, or why that's a good thing for us? But what Zaid said is correct.
Is it so they can have more hemoglobin and carry more oxygen around the blood?
Exactly. That is the other reason. Exactly. That is why our red blood cells don't have a nucleus, but the non-mammalian vertebrates, theirs do. And again, this could be because of the different metabolic needs. Okay, so mammals, our red blood cells are, of course, unnucleated, meaning that we don't have a nucleus. They are a biconcave disc, as they'd mentioned, and we don't have as many organelles. And this is, of course, to carry more oxygen, as Darude mentioned. So amphibians, they have the largest red blood cells that you can see right here. These are the largest red blood cells, and this may be because of their large genomes. Large genomes meaning a large size of the genes, basically. The endothermic amniotes, they normally have smaller erythrocytes. Now, the functionality. So, the gas transport efficiency is determined by the size of the erythrocytes. Okay? Usually, the bigger the erythrocyte, the more efficient the gas transport could be. The mammalian erythrocytes are a biconcave shape because of the enucleation, of course, where they don't have their nucleus. And this, of course, increases the surface area for gas exchange so that they're able to take in more oxygen and take in more carbon dioxide to either pass the oxygen, of course, to the cells and the tissues that need them, or to pass the carbon dioxide out of the body. Okay. Now, let's move on to the next slide. So this is to do with the development of the heart. Now, this is also very good to remember or to learn for anatomy. So we are going to discuss it very quickly. Just bear with me. Okay. Right. So the heart, it basically develops from the mesoderm, from the germ layer, the mesoderm. It will develop 18 to 19 days after fertilization has first happened. Now, this might sound a bit complex, but don't worry, it won't be that hard. But the mesoderm is divided into separate areas. It has 3 parts. The mesoderm has got the other parts here, actually. The parts are not labeled here, but the mesoderm is divided into the paraxial mesoderm, the intermediate mesoderm, and the lateral plate mesoderm. I do have another picture of that in the section where we're talking about the excretory system, the development of the excretory system right here. Okay, so This middle portion, of course, is our mesoderm. Okay, this area here on the top is our ectoderm, and then on the inside we have our endoderm. But anyway, this middle part is our mesoderm, and that's what we're talking about at the moment. So we said that the mesoderm is divided into 3 parts. We have our paraxial mesoderm right here. We have our intermediate mesoderm in the middle, and then we have our lateral plate mesoderm on the side. The heart, okay, develops from the lateral plate mesoderm. Let me go back to the pictures. Okay. So the heart develops from the lateral plate mesoderm. Now this lateral plate mesoderm, it basically is going to develop these cavities or these holes that are going to form the intraembryonic cavity. As you can see, these are holes, okay? It's forming the holes, which are going to be the intraembryonic cavity, okay? So this forming of the intraembryonic cavity is going to split the mesoderm further into the somatic region at the top and the splanchnic layer at the bottom. Okay. Now the heart is going to develop in the splanchnic layer at the bottom. Okay. And what it's going to do first is it's going to start off as clusters of cells. And the endoderm, which is at the bottom, of course, this blue part is the endoderm. The endoderm is going to stimulate these clusters of cells to become what we know as blood islands. Okay. Now, these blood islands are later going to become vessels in a process called vasculogenesis. They're going to become the blood vessels. Okay. And they are also going to form the heart tubes. They're going to form 2 heart tubes. Okay. These 2 heart tubes are going to form one primitive heart because they are going to fuse together. So we have our 2 heart tubes, they will come together and they will form the earliest structure or the most primitive structure of the heart. Okay? And these are the main 5 regions. And then over time, as the heart is developing, it's going to start to bulge out, especially at the bulbous cordus region and the ventricle region. It's going to bulge out, and then the atrium and the sinus venosus, eventually it's going to fold in on itself. It's going to form this S shape. And it's going to form this S shape so that the heart is more compact and so that it can fit kind of better within our chest space, basically. And it's going to form this shape right here. So this is the earliest form of the heart. Okay. Does anybody have any questions before we move on to the last slide of this syllabus point? No questions? Okay. Now, the aortic arches, what happens to them? Well, the third arch will become the internal carotid arteries. The fourth arch is going to divide, and the left half is going to become the aorta, whereas the right half is going to become the right subclavian artery. So right here, the right subclavian artery. Okay, and then arch 6 is going to form the pulmonary trunk right here at the bottom, which is going to split into the right pulmonary artery and the left pulmonary artery. Then all the other arches, other than these 3, are going to disintegrate. They're going to degenerate. So this is in human development, by the way. So in human development, we only have these 3. Okay, all the other arches, so arch 1 and 2, they are gone. Okay. All right, so that was the end of the circulatory system. We're going to go into the respiratory system, but I'm going to take a quick break of 2 minutes. Okay, and then we're going to start. So in 2 minutes, we're going to start again, and we will do our respiratory system, and then we'll do our excretory system, and then finally our finally our reproductory. So we basically have 3 systems left to go. Okay, but I'm taking a 2-minute break. Okay, guys, I'm ready to start again. All right. So, we are talking about the respiratory system now. Can you all see the screen? Yes? No?
Yep.
Yeah, great. Okay. So, the respiratory system, so vertebrates basically, they have their gills and they have lungs, which they use for respiration. Of course, not all vertebrates have gills and lungs, but usually those are the two organs which they use for respiration. Both of these come from the pharyngeal endoderm. Okay. The evolution of respiratory system involves these two organs, as we said, the gills and the lungs. So, initially, the chordates didn't really have a respiratory system. So, the protochordates, which are the earliest chordates, for example, the lancelets, they used to rely on their body surface to absorb in oxygen. They didn't really have gills, so they used sort of gas exchange in their pharyngeal slits that we mentioned before in order to sort of remove the waste gases. Okay, so the gills first appeared in the agnathans, which are, of course, the jawless fish. They developed gills in the pharyngeal slits and converted them to what is known as the branchial slits. Every time we're talking about fish and we're talking about the pharyngeal slits, we're going to say the branchial slits, okay? Because in fishes, they're called branchial slits. The modern agnathans, they have specialized gills. Now, the gills and gill slits in the cartilaginous fishes, so gnastostomes, which are fishes that have jaws, they have numerous gills which have blood, which basically supply, they have a supply of blood, basically. Remember when we were speaking about the circulatory system and we were talking about the afferent and the efferent branchial arteries? Well, that is the blood supply that we're talking about here when we're mentioning it to do with the gills. So the development of the gills correlates with the main blood vessel, the development. Okay, so how does the blood flow through the gills? Well, the venous blood comes from the ventral aorta and enters the afferent branchial arteries. Okay, where is it? So right here. Comes from the ventral aorta and enters the afferent branchial artery. There. Okay, now oxygenation occurs in the gill capillaries where they're receiving the oxygen. The oxygenated blood exits as the efferent, it exits basically through the efferent branchial arteries and joins the dorsal aorta and the carotid arteries, and then eventually, of course, goes to the cells and the tissues that need them. Now, cartilaginous fishes, they take in water through their mouth or another structure which they have called the spiracle around their mouth area. They take in this water, and then bony fishes, they use opercular movements for gill ventilation. Now, opercular movements is to do with the opercular valves which they have right here in their necks, okay? So, usually what happens is the fish is going to open its mouth and it's going to take in water. When it does this, the opercular valve is closed, as this allows for the fish to take in oxygen from that water through the gills, right? Now, when the fish doesn't need to take in any more water, it closes its mouth and then opens the opercular valves in order to remove the water now that it's taken the oxygen out of it, okay? So this is water that it doesn't need anymore. So that is how the opercular movements work. The amphibians, they use external gills for respiration. Their gills regress during metamorphosis, meaning that they basically get rid of their gills as they become adults, as we mentioned before. And some parts of these gills will form the parathyroid glands. Then in amniotes, gills are absent in the amniote embryos, as the pharyngeal slits, which would have formed the gills later, will evolve into other structures instead of becoming the gills. Because amniotes, of course, they don't need these gills anymore, as they're not going to be living in water. Okay, so the fate of lungs in bony fishes. So we mentioned swim bladder before. And I mentioned something to do with the swim bladder before. And what I told you guys was that I said that bony fishes, when we were talking about their skeleton, bony fishes have a skeleton made out of bone. Okay? Whereas cartilaginous fishes, of course, their skeleton is made out of cartilage. Okay, so the bony fishes, they're able to have a skeleton made out of bone, where bone is, of course, more heavy, right? And this is because these fishes, these bony fishes, normally have a structure called a swim bladder. Now, this is the placement of the swim bladder. This is where you can find it in these bony fishes. Now, this swim bladder, you can think of it as like a bag that contains a lot of oxygen. Okay, now this bag that is containing a lot of oxygen is basically making it possible for these fishes, these bony fishes, to stay afloat in the water. Okay, but cartilaginous fishes, they don't have this swim bladder. What would happen if cartilaginous fishes were made out of cartilage skeleton instead of a what would happen if their skeleton was made out of bone? If the cartilaginous fishes, if their skeleton was made out of bone instead of cartilage, what would happen to those cartilaginous fishes?
It would be too heavy.
It'd be too heavy, exactly. And these cartilaginous fishes, we said that they need to be effective predators. They need to be able to swim properly. They need to be able to chase after their prey and stuff like that. So for that reason, they don't have a skeleton made out of bone. Their skeleton is made out of cartilage instead. And then the bony fishes, they can afford to have a heavier skeleton. Reason being, they are usually smaller, okay, in size, and also because they have this swim bladder, which keeps them afloat, basically. Because, as we said, the swim bladder is like a bag that has oxygen, which promotes buoyancy, which is like that, floating in the water. Okay, so the evolution of the nasal passages for respiration. So in lungfishes and amphibians, first of all, they had two sets of nostrils, okay? So they have the anterior nostrils, which are at the front, and then the posterior nostrils, which are at the back, okay? So in order for them to start to use their nostrils for respiration, actually, because before they were only using it for olfaction, which is smelling. They were only using it for smelling, but they need to start developing it for breathing because, as you know, we're going to go from animals which are using gills for respiration to animals which are going to only use lungs as they're going to become land animals. Okay, so what's going to happen, or what happened, was that the posterior nostrils, which were at the back, they basically moved into the mouth. Okay, so they moved from being at the front of the face, near on top of the mouth, to actually coming into the inside of the mouth. Okay, so this connected the nasal passages to the respiratory tract and made it possible for them to now be able to breathe. Okay, to breathe through their nose or to breathe through their nostrils. Okay, now the adult amphibians, they have a primitive they've got primitive sac-like lungs. They formed a septa, which basically divided their lungs. So from the bronchioles, it divided the bronchioles. So there was a separation between the bronchioles and the alveoli. Okay, and this was, of course, to help with the gas exchange, because as you know, the gas exchange primarily happens, of course, in the capillaries in the alveoli. We also mentioned before last week that the amphibians, they do a lot of their gas exchange on their skin also, okay? But they need to keep their skin moisturized in order for this gas exchange to happen. And then, of course, as we just mentioned, these amphibians are also able to take in air through their nostrils, okay? So they're able to basically breathe in air this way too. And these nostrils actually can be closed. If they want their nostrils to be closed, they can close them, basically. So their respiratory pathway, their air goes through the oral cavity and crosses path with the food and water. This is different to mammals and different to humans. As you know, we have two separate tubes. One for breathing, which is our trachea, our windpipe, and then, of course, one for our food. Whereas the amphibians, they don't really have this because they are smaller animals and because of their pharynx and their glottis. So their pharynx has slit-like openings, which are referred to as the glottis, and this is basically the structure which allows for their air to go into the larynx. Their larynx then, of course, is the structure which has the vocal cords that allows for sound production. Now, the amphibians, they've got something called buccal pumping, okay? Now, you may have seen videos of frogs doing this, or you may have seen this in person, I don't know. But basically frogs, when you look at them for a while, what they do is they're just basically sat there or stood there or whatever, and you will see actually that the oral cavity, so just underneath their neck area, is basically filling up and then deflating like a balloon, you know, filling up and deflating like a balloon. And this is basically buccal pumping. It is the up and down movement of the oral cavity floor, which is controlled by the hyoid apparatus. And this is essentially how they breathe. Okay, so in inspiration, the buccal cavity expands and gets larger as they draw air into the buccal cavity. Okay, then in expiration, the buccal cavity will contract, the air will be forced out of the nostrils, and then the air is pushed into the lungs. Okay, so this is how they breathe. Now in reptiles, actually, reptiles, they don't have this buccal pumping, and reptiles and other amniotes, we have intercostal muscles, which are actually going to help with this. Okay, our intercostal muscles are the structures which are helping with our inspiration and our expiration. For this reason, amphibians, which don't have the intercostal muscles, they usually also either they have ribs or they have a very small, like a small number of ribs, okay? Because their ribs are not helping with their respiration. Whereas for us, of course, our ribs help us with our respiration, as do our intercostal muscles. Okay, so intercostal muscles for the first time were seen in the amniotes, and the intercostal muscles, as I said, replaced the buccal pumping. The septa became better developed, so there was a better separation between the bronchial and the alveoli, which would increase the gas exchange surface area, which would happen in the alveoli. The amphibian lungs and the reptile lungs are kind of structurally similar, but there are some improvements in the reptile lungs. Okay, so the secondary palate that we mentioned that the mammals have for the first time can also actually be seen in the some reptiles. So it can be seen in crocodiles and sea turtles. And basically the importance for this is that you know, when they're diving deep in water, they will still be able to breathe while their mouth is full of water, because this secondary palate has been pushed back. It's been pushed to the back. Okay, meaning that, you know, those nostrils that they had that we were talking about, they are also in the back, and in this way they're able to breathe. They're not drowning whilst they're in water. And this is specific to the crocodiles and the sea turtles. And the conne is right here. So it's the conne, which is in the secondary palate, which is going to move backwards. Okay. Now mammals, they developed the respiratory turbinates, and this would basically help them reduce water and heat loss. These turbinates are also known as the maxilloturbinate bones, and these are only seen in mammals. Okay. Now, the larynx also became more sophisticated or more developed in mammals because, as you know, we use our vocal cords in order to communicate, and so do other mammals. For example, monkeys, apes. The epiglottis and the thyroid cartilage developed, okay? And the epiglottis and the thyroid cartilage, basically, they stop food from going down our windpipe or our trachea as we are swallowing, okay? As otherwise, if the food was to go through our trachea or our windpipe, then of course we'd be choking and we would not be able to get any air supply and we would die, right? Mammals also developed the diaphragm for the first time, and this helped with the lung ventilation and helped in breathing efficiency, basically. Now, the development of the respiratory system. So first, the development of the gills, and then we're going to talk about the development of the lungs. Just bear with me. Okay. So the gills in the vertebrates, they develop through a coordinated process that will start with the forming of the pharyngeal pouches, which will come from the endoderm. Okay, these pouches are going to grow outwards, and then they're going to interact with the mesoderm that is above them, and then eventually it will fuse with the ectoderm, which is the outermost surface, and this is going to create the gill slits. All right, then there's going to be an epithelial lining which will be covering these slits, and this is going to form the gills themselves, and this will allow these organisms to basically extract oxygen from the water. So just to round up again, we said that the gills are going to form from the endoderm of the pharyngeal pouches. It's going to basically pierce through the it's going to pierce through the mesoderm, and it's then going to interact with the ectoderm, which is all the way at the top. This ectoderm is going to form the epithelial layer, which will cover the gills and essentially actually form the gill slits. Okay, and that is the development of the gills. Now, for the lungs, they come from their own initial structure, which is what we mean by anlage. Okay, they come from their own initial structure and they form as outgrowths of the foregut. Okay, we mentioned the foregut earlier, and we mentioned how the foregut can actually also become the stomach. The foregut can also start the development of the lungs. Then the lung buds are going to grow and branch extensively and form the complex network of airways. Okay, and then these air spaces are then going to be subdivided by the alveolar septi, and this is known as septation or alveolarization. And this is, of course, basically the splitting up between the bronchioles and the alveoli. Okay, these alveoli are important, of course, for gas exchange, and they allow the lungs to efficiently transfer oxygen into the blood and to remove the carbon dioxide from it. And that was the point about respiration. Any questions, or is anybody confused? Does anybody need any help with anything that we discussed in respiration? No? Okay, then we're going to go into the second, not the second, but to the next part, which is syllabus point 72. So this is the phylogeny and the development of the excretory system. So again, we first start off by talking about what is the excretory system and what is it important for. Well, of course, it is important for removing waste products, but also for detoxifying, okay? So there's something I want to tell you that has to do with biochemistry a little bit, but it could help you also understand this point a little bit better, maybe. So our body, we basically need to make a lot of energy, and we make energy in the form of what? What is our main currency of energy that the mitochondria makes?
ATP.
Yes, ATP, exactly. So we make energy in the form of ATP, and we are normally, our body's preferred way of doing this is by breaking down carbohydrates, okay? Because breaking down carbohydrates gives us glucose, all right? But there are points where we may not eat the correct food, right? And we may not be eating carbohydrates or as many carbohydrates as our body needs. Or there are points where somebody might be starving and they may not be eating anything at all. In these cases, the body is a very interesting thing because it does its best to survive, okay? So if it realizes that you're not eating any carbohydrates, it will start to break down amino acids, okay? And it will start to break down lipids. And these are lipids which it stored basically for you. So it will break down these amino acids, and when it breaks down the amino acids, we release something called ammonia. Okay. Now this ammonia is very, very toxic for our brain, and it's very toxic for our other tissues also. So the excretory system, it works really hard to remove this ammonia, okay, so that we don't develop certain neurological issues. Okay, so we remove these nitrogenous waste products, which is, for example, ammonia, and this will help to detoxify the body. And we're basically, when we are getting this, when we are breaking down the amino acids and we are getting ammonia, we are going to, as mammals, as humans, we are going to convert this ammonia into urea. But there are some species which convert the ammonia not into urea, but they convert it into uric acid instead. And these species are the reptiles, okay? And we'll talk more on that a little bit later. So the main point of excretion is to remove the nitrogenous waste products. It's also to regulate the osmotic pressure, okay, to make sure that we have enough water and enough salt, because if we have too much water or we have too little water, then our cells can either swell up with too much water and burst, or our cells can shrivel up when we have too much salt and less water, and the cells can basically become dehydrated, which of course is not ideal for us. Okay, so the osmotic pressure is also controlled by the excretory system, and the acid-base balance is also of course controlled by the excretory system, where different parts of our body need to either be acidic or basic, depending on what kind of enzymes we have there and depending on what kind of processes are happening in those areas. So our kidneys are able to actually buffer our blood to maintain our pH, our blood pH, basically. So this is the excretory system, of course. It consists of our left and right kidneys, our ureter, our urinary bladder, our ureter tube, this tube right here, our urinary bladder, and our urethra. Okay. Now, the parts of the mammalian kidney. So we've got the cortex, we've got the medulla, and then we have the pelvis. So this area here is the cortex. So this pinkish area here is the cortex. It has the renal corpuscles and the convoluted tubules. We're going to talk about the renal corpuscles in the next slide. Then we have the medulla, which is this area here. The medulla contains the loops of Henle and the collecting ducts that are going to form the pyramids. Okay. Then we have the renal pelvis, which is this structure right here, which is going to connect to the ureter. Okay. And this ureter, of course, is the area that's going to collect the urine. And then when the ureter collects the urine, it's going to send that urine, of course, to the urinary bladder, where it will be stored until it's full, and then you're going to excrete it through the urethra. Okay, so the nephron is very important to talk about for this syllabus point, because the nephron is the most basic unit, basic structural and functional unit of the kidney. So if you just basically dissect an area of the kidney right here, you would basically find a nephron. The nephron consists of a glomerulus, which is basically an area that has a lot of capillaries that filter the blood. And then the nephron also has got the tubules, okay, the renal tubules, and these tubules are important for reabsorbing and for secretion. Okay, now this is reabsorbing of salts, of certain nutrients, etc. Then the components of the nephron. So the nephron's got the Bowman's capsule, which basically encloses around the glomerulus and forms the renal corpuscle. Then the renal tubule right here, okay, starts as an extension of the Bowman's capsule and opens eventually into a collecting duct, okay? This collecting duct is not considered to be part of the nephron. Okay. So now more about the renal corpuscle. So we basically said that the renal corpuscle, actually it's also called the Malpighian corpuscle, and it consists of the Bowman's capsule and the glomerulus together. We said that the Bowman's capsule is basically encapsulating the glomerulus. It's basically just an area which is surrounding the glomerulus. We said that the glomerulus is basically a bunch of capillaries all together, which are going to filter the blood. So the afferent vessels, okay, can be found in the glomerulus, and the efferent vessels. The afferent vessels are going to supply blood to the glomerulus, whereas the efferent vessels are going to drain the glomerulus, and they're going to contribute to increasing the blood pressure in the glomerulus. This increase of blood pressure is going to be the driving force, which is going to allow the Bowman's capsule, as you can see, it's got cells right here. It's going to allow these Bowman capsule cells to filter this water and solutes. Okay, and then of course this is going to send that to the tubules, to the renal tubule, and then eventually to the collecting duct. Okay, then we have something called the renal portal system, which is important in and can be found in cartilaginous fishes and birds, up until birds. So all the vertebrates except for mammals. Mammals do not have the renal portal system, but all the other vertebrates, they do. Now, this renal portal system, essentially what it does is that it basically takes blood from the hind legs or the tail, so the posterior part of the body. It takes blood from there and then essentially sends it to this area, to the renal portal area, before this blood goes back to the heart. Okay, so in this way, it basically supports the nephrons in low arterial pressure when there's low arterial pressure and allows for the nephrons to work even if the glomeruli are basically reduced in this species or they are absent. Okay, so they are absent in mammals. Mammals, they don't have this renal portal system because we have kidneys that are highly efficient at filtration. And also our kidneys are very good at reabsorption. Okay, so in this way, we don't need this extra feature. We don't need this renal portal system, but other animals which are less advanced than we are, they still need it. Okay, and they still need it in order to filter that blood that is coming from the posterior parts of the body. Okay, now the phylogeny of the excretory system. So first we have to understand what integrated and non-integrated nephrons are. So integrated nephrons, they have a glomerulus that is enclosed by the tip of the renal tubule, which will form the Bowman's capsule. In other words, integrated nephrons have a Bowman's capsule around the glomerulus. So that is B right here. So we can see, so basically we just said that the tubule is enclosed around the glomerulus, which forms the Bowman's capsule right here. This is an integrated nephron. The non-integrated nephrons, they don't have this, meaning that they are lacking the renal corpuscle. We said that the Bowman's capsule and the glomeruli together is referred to as the renal corpuscles. But the non-integrated nephrons, they don't have that renal corpuscle. They have a glomerulus that will filter the fluid directly into the colon. The colon is like a space or cavity, which is basically found in the body. Okay, so it will directly just send the filtered blood or the filtered liquid directly into the body cavity. Okay. The renal tubules are then going to draw fluid from the colon through a funnel-shaped ciliated proximal ends called the nephrostomes. So after the blood is filtered, it will be sent to the colon. Then it's going to be drawn up by the nephrostomes right here. These nephrostomes are ciliated, meaning that they have cilia, which is going to push that liquid up into the tubule, then down into the tubule, and eventually into the collecting duct. Okay, so that is the difference between the integrated and the non-integrated nephrons. Okay, now there's 3 stages of the kidney evolution. We're going to start with the pronephros, which is all the way here on the left. These are the pronephros. They develop in the front part of the body. They have non-integrated nephrons, meaning that they have basically this structure. This is the non-integrated nephrons. And they filter the liquid directly into the colon, which was the body cavity. Adult lancelets, which are the early chordata, use pronephros. They have the pronephros. Then the mesonephros developed behind pronephros. In some species, the pronephros degenerated. In some species, they still kept their pronephros as they developed their mesonephros. This mesonephros has got both integrated and non-integrated nephrons, and anamniotic vertebrates use this as adults. What are anamniotic vertebrates? Can somebody tell me? What are anamniotic vertebrates?
Vertebrates that don't lay eggs.
Vertebrates that don't lay eggs. Not really. So amniotes are the first animals which were able to lay eggs on land, right? So these are the reptiles and the birds. And then anamniotes are the opposite. Anamniotes are the animals which are living in water. So this would be the fish and the amphibians. So the fish and the amphibians, they developed the mesonephros, okay? Especially in their adult forms. Then we have the metanephros, and this would be now the compact kidney that would now have thousands or millions of integrated nephrons. So these ones, integrated nephrons, in a branched arrangement. So we'd have a lot of nephrons within these kidneys. And these metanephros are now used by amniotes, which are, of course, the mammals, reptiles, birds, and it's basically developed more as they become adults. Okay, so excretion in fishes. So fishes, they excrete the nitrogen as ammonia through their gills. The ammonia, of course, as we said, is toxic, but it diffuses quickly through their gills and causes them no harm. These gills, they retain the excretory function even as they become air-breathing lungfish. Okay, they're still able to excrete this nitrogen. And the function of the kidneys in the fishes primarily is for osmoregulation. Okay, and this is essentially how osmoregulation works. When you have a low amount of water in your blood, this will send a message to the brain, and the brain will start to release antidiuretic hormone. It will produce a lot of antidiuretic hormone. And this antidiuretic hormone is basically going to send a message to the kidneys where it will tell the kidney, hey, we don't have enough water in our blood. Can you start to take in more water from the blood? Like stop excreting the water and keep more of the water in the body. Now, because more of the water is kept in the body, this means that there's less urine that is released, and the urine that is released is going to be concentrated because the water that would have been in it and would have diluted it otherwise is not there, because the kidney has been told by this antidiuretic hormone to, reabsorb that water into the body. Then eventually the water content in the blood will be normal. And then if you have too much water, or too much water in the blood, the brain is then going to release less antidiuretic hormone. This is going to send a message to the kidneys, which is going to tell the kidneys, hey, you're taking in too much water, you're reabsorbing too much water, we need you to release more water. Okay, so because of this, there's going to be a higher output of urine. You're going to produce and release more urine, and then the urine is going to be more diluted because, of course, now there's more water that you are releasing, basically. So this is what fishes do, and this is also, of course, what we all do, actually. But this is what osmoregulation is. Okay. Now, the excretion on land for amphibians. So kidneys replace the gills for nitrogenous waste excretion. As you know, amphibians, when they became adults, they lost their gills. The ammonia becomes processed to urea, which is similar to us as humans. And this urea is toxic, but it is less toxic than ammonia. Okay. And then the amphibians, for the first time, developed a urinary bladder, which would control their urine storage and their urine removal. Now, the purpose for the urinary bladder is basically so that they can reabsorb water from their bladder whenever they have no water, basically. When they are lacking water in their body and they're not really able to find a water source, they will reabsorb water from their bladder. Okay. Now, reptiles and birds. So reptiles and birds, actually, they convert their nitrogenous waste to uric acid instead. Instead of urea, they convert it to uric acid. And this is because when they are doing this, they basically don't need to release as much water when they are releasing this uric acid. And the whole reason for this is because we said reptiles and birds, they're going to be the first animals which are completely independent of water. They're going to move away from the water sources, and they don't know when their next source of water is going to be. So they're really trying to retain that water in their body. So they're doing anything and everything that they can, which allows them to minimize the amount of water that they're going to lose. And one of those things is to convert their ammonia into uric acid, which needs less water for them to basically pass it out. They also excrete a dense semi-solid white paste of uric acid crystals with their feces, which again helps them basically retain their water. Okay, and they have a small glomeruli filters which use just enough liquid in order to transport this uric acid to the cloaca, which is their anus region. And then most of the birds have lost their urinary bladder to reduce their body mass. Why would birds need to lose their urinary bladder? Why don't birds, most of them, why don't they have a urinary bladder? Think about it. What do birds do that other animals don't do?
Fly.
Yes, exactly, they fly. So when they're flying, of course, they have to act against gravity, right? So if they had like a sack containing liquids, a sack of urine, basically, this would basically reduce their ability to fly, or reduce their ability to fly high. right? So for this reason, most birds, they lost this urinary bladder, which helped to reduce their body mass, which meant that they'd be able to fly better, or to fly at all, basically. They'd be able to fly because they lost the urinary bladder. Now, in mammals, for urea excretion, mammals convert nitrogenous wastes to urea, as we mentioned, and this urea has to be dissolved in water and is then released as urine, okay? The early amniotes also probably used urea, and the mammals, basically, they branched off and became their own species before the reptiles and birds started to switch to uric acid. And as we said, reptiles and birds switched to uric acid so that they would need less water in order to remove this uric acid from their body. Now, the filtration requirements. So intensive filtration and adequate glomerular blood supply is needed in mammals because, of course, as we said that these mammals or amniotic animals in general, they are requiring more, not amniotic mammals in general, but birds and mammals, basically. Of course, they have a higher metabolic need, okay? And because of this, they are basically, they need to filter their blood, and yes, they need to filter their blood more. And in order to filter their blood more, they need to have more glomeruli, basically. The renal portal system in mammals was lost. So even if mammals had wanted to change the ammonia into uric acid, this would not be possible because we don't have the renal portal system. Okay, so we lost the renal portal system as we stopped having to, basically, we didn't need to make uric acid anymore. We started making urea instead. Okay, so the urine concentration ability in mammals. So most mammals are able to produce urine 3 to 4 times that is more concentrated than our blood plasma. The desert species, so animals which live in the desert like kangaroo rats, are able to produce 17 times more concentrated urine. Why would they need to produce concentrated urine, or why would their urine be concentrated? Well, this is because they are trying to avoid dehydration. They're trying to make sure that they're only removing water that they may not need, whilst at the same time trying to keep the water balance within our bodies constant to avoid dehydration. Okay, and this is mainly because these mammals now, they are endothermic, as we said, and they have intense metabolism where they require a lot of food and a lot of energy, and they don't have the renal portal system, which makes them vulnerable, which makes their nephrons vulnerable to dehydration. So due to this, they produce a lot of concentrated urine rather than diluted urine. Okay. Now, the excretory system and its development. So the excretory system develops from the mesoderm, specifically the intermediate mesoderm, which is, of course, in between the lateral plate mesoderm and the paraxial mesoderm. Okay, the key structures which form would be the nephric or the Wolffian duct, which we're going to mention in the reproductive system. But the nephric Wolffian duct, is formed by the dorsal part of the intermediate mesoderm. The dorsal part of the intermediate mesoderm means the back part of the intermediate mesoderm, and then it's going to develop into the collecting ducts and the ureter. So if we just look again, the collecting duct is right here. Okay. And the ureter is right here. So the Wolffian duct is going to make the ureter and the collecting duct. Okay. Then the nephrogenic cord, this is going to give rise to the nephrons. As you know, we said that the nephrons are the most basic structural and functional parts of the kidney, correct? And then the glomeruli is, of course, responsible for blood filtration. So the intermediate mesoderm gives rise to the pronephros, the mesonephros, which are going to give rise to the gonads. These are the testes and the ovaries. And then the mesonephros are going to give rise to the metanephros, which are going to give rise to the kidneys. Okay, so that is the excretory system. Any questions before we go to the last system? No? Okay. So this is the last part of phylogeny and development that we will be, that is in the syllabus, and of course that we'll be discussing today. So this is to do with the reproductive system. So of course, for the reproductive system, you need to be able to give an overview of which organs are involved. And of course, as I said, when you're giving this overview, you're going to talk about it as in relation to humans. Okay? So the overview, first of all, when it comes to the phylogeny, so of course we have anamniotes and we have amniotes. Anamniotes are animals like the fish and the amphibians, okay, and we're going to talk about them first. So their spermatogenesis is cystic, meaning the formation of the sperms happens inside of cysts, okay? You can think of these cysts as little bags which are inside the Sertoli cells. Okay, now these cells, these spermatogenic cells, are connected by cytoplasmic bridges, meaning that these cells are connected together, but through their cytoplasms, basically. Okay, and this allows them to have synchrony, meaning that this allows these cells to grow at the same time. Okay, and as they're growing, they are growing inside of the cysts, which are the bags that we mentioned, and these cysts are eventually going to rupture, and when they rupture, they will release the spermatozoa. So they're going to release a lot of them at one time, and this happens in the fishes and the amphibians. Okay, then the amniotes, which are of course the reptiles, birds, mammals, they have the seminiferous tubules. Okay, the testes are organized in these narrow, long seminiferous tubules, and it's composed of Sertoli cells and the germ cells. And this structure allows for high productivity and continuity in spermatogenesis. And as you know, spermatogenesis, when we're talking from a human aspect, is something that happens, of course, in steps. So in these cells, they're not like the cells in the anamniotes, where the cells are connected cytoplasm to cytoplasm, or they're sharing a cytoplasm. They are actually separate cells, which are going to develop bit by bit and eventually going to be released. Okay? Now the ovaries. So most vertebrates, they have large ovaries because they have eggs which have large or a large amount of yolk in them. As we mentioned before, the yolk is there to provide nutrients to the embryo. Okay. Mammals usually have tiny and yolkless eggs. This is because our placenta developed. And the placenta basically has the same function as the yolk, where the placenta, of course, is providing nutrients directly from the mother to the embryo. Okay, and because of this, these eggs that these mammals are now producing, now that they have the placenta, these eggs can afford to be smaller and can afford to have less yolk or no yolk at all. Okay, so because we're going to produce smaller eggs, we're also going to have smaller ovaries. Okay, so the phylogeny of the female genital tract. So in non-mammalian vertebrates, the female genital tract comes from the Mullerian ducts. Okay, so these are the Mullerian ducts. And special parts of the the Mullerian ducts can later form the oviduct. It can also be called the oviduct. This oviduct can be referred to as the uterus later on when it becomes specialized. And then the oviducts, which you can find in fishes and amphibians and reptiles, are paired and they open separately into the cloaca, which is the anal region. Okay, and then eggs are released into the body cavity. So the eggs are released by the gonads into the body cavity and enter into the oviducts. Okay, and in internal fertilization, what happens, of course, is that the sperm cells will travel into the proximal part of the oviduct, so basically to the upper region here. Now, later on, so basically going from the non-mammals, in mammals, what happens is the Mullerian ducts, they're going to eventually fuse together. And when they fuse together, we're going to get the uterus, like this. Okay. Now we have to talk about certain animals. So monotremes are animals like hedgehogs. They lay eggs and they have reptile-like female genital tracts. Then we have marsupials, which are like kangaroos, for example. And these marsupials, they have fully paired female genital tracts, which are adapted for viviparity. Viviparity basically means giving birth to live young, like containing your embryo in the uterus, growing it in the uterus, and then giving birth to live young. So because of this, these marsupials, they have fallopian tubes, they have a actual uterus, and they have a vagina for delivering the offspring. Then we have placental mammals, which is the difference actually between marsupials and mammals, or mammals that have placentas, because marsupials, they don't have a placenta, okay? Whereas, of course, placental mammals, they have a placenta. They normally have paired oviducts and a single vagina. Their uterus can be duplex, so it can be fully paired, or it can be simplex, meaning that the uterus is fully fused, or it can be a combination of both, where it is fused and also is fully paired. Simians, which are basically primates, for example, have a simplex uterus, meaning that our uterus, like humans, for example, monkeys, for example, our uterus is fully fused. So the Mullerian ducts, they have fully come together, fully fused, and formed our uterus. Okay. Now, how does the reproductive system actually develop? So in organogenesis, which is of course the formation of organs, the gonadal or genital ridges, they form. And they form basically quite close to, in front of, and next to the nephrogenic cord. Remember that we said that the nephrogenic cord is going to give rise to the nephrons, which are of course the structural and basic units of the kidneys, right? So the gonadal ridges are going to form right next to them. Okay, and the gonadal ridges are right here. You can see them right here. Okay. All right. So it's made out of the epithelium. So the gonadal ridges, they're made out of epithelium and the underlying mesenchyme of the intermediate mesoderm. So again, if you're asked where does the reproductive system develop from, well, it forms the gonadal ridges, and the gonadal ridges come from the intermediate mesoderm, which is similar to the excretory system. The excretory system also develops from the intermediate mesoderm. Okay. So now the reproductive system development in terms of the, in regard to the primordial germ cells. So the primordial germ cells are also known as gonocytes, and they are the earliest gamete precursors. They originate in the epiblast, right, which is here. And if you remember, the epiblast is also going to form the embryo later. So the gonocytes, they're going to form in the epiblast, and then they're going to move down to the yolk sac, which is going to basically protect them from any further differentiation. Because as you may know, when the embryo development is happening, there's going to be a lot of cell-to-cell induction, where some cells are telling other cells, hey, you need to go over there because you're going to become this, and you need to go over there and you're going to become this. So the gonocytes, they're determined quite early, and they're protected by the yolk sac so that other cells don't confuse them and tell them, okay, I know that, we said that you're going to become the sperm or the egg cell later, but actually what I need you to do is I need you to go over there and become this. It protects them from that, basically. Okay. So the migration. So from the yolk sac, these gonocytes are then going to split into the left and right populations, and they're going to migrate dorsally, meaning they're going to move to the back where the newly formed hindgut is. Okay, so this yellow structure is the hindgut. So the cells, the what's it called, primordial cells, the sperm cells or the early sperm cells and early egg cells, they're going to move from the gonadal ridges, from the yolk sac, and then they're going to go into the hindgut. Okay, so these blue structures here. Okay, and then they're going to settle in the genital ridges and develop into the true gonads. So this would be either the ovary or the testes. Now, the sex determination is also important to mention with this syllabus point. So early gonads have the potential to become either ovaries or testes, and this development is influenced by somatic cells, which are other body cells, which are found in the gonadal ridges, and it's also influenced by the sex chromosomes. So in XY individuals, which will of course be males, the XY chromosomes, the pre-fertilized cells, which are going to become the fertilized cells, are going to form the testis cords around the germ cells. And then Leydig cells, very early Leydig cells, are going to differentiate around these cords. And later, these Leydig cells are going to release and produce testosterone. the satelli cells are going to secrete anti-Müllerian hormone. And this anti-Müllerian hormone is going to basically get rid of the Müllerian duct so that the male is not going to form the female genital tracts, basically. This is going to drive the male reproductive system. Give me 1 minute. Okay, sorry, I have to take my cats to the other room because they just woke up. Okay, but we're almost done. Okay, then in XX individuals, which are of course going to form the females, the supporting cells became granulosa cells instead and formed around the germ cells, and this eventually formed the primordial follicles. Now, the SRY gene is very important for sex determination because the SRY gene can only be found on the Y chromosome. And of course, the Y chromosome can only be found in males, okay? And this Y chromosome would trigger the development of the testes. Now, the SRY gene, it will activate another gene called the SOX9. You don't have to remember that or mention that if you don't want to, but the SRY gene is very important to mention. Okay, so this is further going to promote the testes formation, and it will further also, of course, inhibit the ovaries from developing. Now, the hormones produced by the testes or the ovary will lead to the differentiation of the male or female genital ducts and also the external genitalia, the secondary sex characteristics, and certain brain functions. The male and female embryos both form the wolf. Sorry, the male and female embryos, they initially form both the Wolffian and the Mullerian ducts. If you remember, from the Wolffian ducts, we are going to form parts of our excretory system too. Okay, so in male development, the testosterone influences the Wolffian ducts to become the epididymis, the ductus deferens, and the seminal vesicles, which are all, of course, going to contribute to the release of the sperms. In the female development, when there is no anti-Mullerian duct hormone, when that hormone is not there, this is going to influence the Mullerian duct to continue to form, and it will then form the oviduct eventually, the uterus, and the upper part of the vagina. And I believe we are, I thought that was the last part. Sorry, we're almost at the last part. So the contribution of the cloaca to the genital tract development. So the cloaca, which is like the anus region, is going to divide into the rectum and the urogenital sinus. This division is going to basically form the urethra in the males and the lower portion of the vagina in the females. Okay, so without this division in the cloaca, those two structures, so the urethra and the lower portion of the vagina, would not form. So the testes development. So we mentioned that the satelli cells triggered by the SRY gene are going to stimulate spermatogenic germ cells and secrete the anti-Mullerian duct hormone. We mentioned that the Leydig cells are going to produce testosterone under satelli cell influence and promote male reproductive structures and secondary sex characteristics. Now, something to mention in evolution that is specific to mammals is that the testes, they descend into the scrotum through the inguinal canal because of a need to have a lower temperature for spermatogenesis. So basically, in spermatogenesis, the conditions are very specific. They can't be exposed to too much heat, for example, when the sperm cells are starting to develop and become sperm cells. And as you know, the sperm cells, they start to develop and become sperm cells in the testes. So initially, what would happen is that the testes, they may have been further up in the body, but then in mammals, over time, of course, in puberty, the testes, they go down into the scrotum, right? And this is usually a permanent thing, and this is to sort of kind of protect the sperm cells from any damages due to heat, okay? And as we said, this is usually permanent, but it can be reversible in some species like rodents, so like rats and mice. This can be reversible in them. I don't know why it's reversible in them, but you don't need to mention why it's reversible in them. You just need to mention this as a whole. Okay. Now the ovarian development. When there is no SRY gene, because of course there's no Y chromosome in females, the germ cells are going to become oogonia, and later they're going to become primary oocytes. The supporting cells are going to become granulosa cells, as we mentioned, and they're going to form the primordial follicles. Then we're going to have Thecke cells, which are basically kind of like the Sertoli cells, but in females. The Thecke cells are going to develop around the follicles, and they're going to become functional at puberty, because as you know, when the female is born female, of course, the ovaries are going to produce they're not going to produce the eggs straight away. It's going to be arrested, right? It's going to be arrested, and then later when it's puberty, when the female reaches puberty, meiosis is then going to actively start. Okay, so the female genital tract development, without AMH, so the anti-Mullerian duct hormone, the Mullerian duct develops into the oviduct, as we mentioned, and the uterus and the upper vagina. This is going to involve the HOX gene, and the HOX gene is basically going to be expressed as this hormone is not there. And it's then going to allow for the Mullerian duct segments to fuse together and to form a single uterus and a upper vagina. And that is the end